Aerobic respiration, Krebs cycle glycolysis has a net result of 2 ATP and 2 pyruvate molecules. Most of the energy from the glucose is still contained in the pyruvate. In the presence of oxygen, pyruvate is transported into the mitochondrial matrix, where it is eventually converted to carbon dioxide. The series of reactions in which pyruvate is broken down into carbon dioxide is called the Krebs cycle, or the tricarboxylic acid (TCA) cycle. This cycle also is referred to as the citric acid cycle.
So, glycolysis result was 2 ATP, 2 pyruvate, 2 NADH. So what we are interested, of course, because those are the stages or the phase 1, the product of the stage 1 will be the reactant of stage 2. The product of stage 2 will be the reactant of stage 3. That means if one of the stages is not working or there is a problem in one of the stages, of course, the whole cycle will be affected. The whole cycle will be affected. So we said glycolysis produces pyruvate, which is 3 carbon molecules. So pyruvate, which is a 3 carbon, will enter into the mitochondria in the presence of oxygen. Of course, specifically in the matrix. Matrix is the empty space of the mitochondria. Pyruvate, which is 3 carbon molecule, will enter into mitochondria, of course, into the matrix. So the reaction that is done by the pyruvate, or the pyruvate is broken down into carbon dioxide, we call it Kreb Cycle, or another name of the Kreb Cycle, TCA cycle, which stands for tricarboxylic acid cycle. Tricarboxylic acid cycle. Tri stands for 3 carboxylic acid, 3 carbon cycle, or another name, citric acid cycle. So those 3 are the same process will happen, or the same reaction will happen, but just they give them different names. Citric acid cycle, Kreb cycle, tricarboxylic acid cycle. Both same reaction, same process, but different names. What is a Krebs cycle? Reactions where pyruvate is broken down into carbon dioxide inside matrix, inside mitochondrial of cells, or specifically inside the matrix. Also called TCA cycle, which is tricarboxylic acid cycle, or simple citric acid cycle. So after glycolysis, aerobic respiration is Krebs cycle. If the cell is respiring aerobically, phase 1 glycolysis. Once done with glycolysis, now they will go to the Kreb cycle. Kreb cycle takes place in matrix of mitochondria in the presence of oxygen. That's why they call it aerobic respiration, or it is known as aerobic respiration. As you can see here in the figure, we start with glycolysis, where glucose broken down into pyruvate. Now pyruvate, which is 3 carbon compound, will enter into the mitochondria, and of course the cycle that is done, aerobic respiration, will first is Kreb cycle, or TCA cycle, or citric acid cycle. Now we are going to see what will happen to the pyruvate. We are going to see what will happen to this pyruvate in Kreb cycle. What will happen to the pyruvate in Kreb cycle?
In the process, a net of 2 molecules of ATP, 2 molecules of pyruvate, and 2 high-energy electron-carrying molecules of NADH are produced. When oxygen is present,
Here they mention that this is a simple product of glycolysis. This is simple. The product of glycolysis to pyruvate, to ATP, to NADH. Of course, if the cell is spiring aerobically, we said it will go to the mitochondria. Specifically, it will enter the Kreb cycle. Of course, pyruvate, which are the 3 carbon, will enter the Kreb cycle. ATP is just energy, a storing molecule, and NADH, they transfer electrons.
Of NADH are produced. When oxygen is present, the pyruvate molecules and NADH enter the mitochondria, and the next stage of cellular respiration begins. The next stage of cellular respiration involves the movement of pyruvate into the mitochondria, where it undergoes oxidation. Each pyruvate molecule is converted into a compound called acetyl-CoA.
So first, even pyruvate, which is 3 carbon compound, it is large enough to enter the matrix. So what happens? It is broken down into 2 carbon compounds, as you can see in front of you. As you can see in front of you here, each pyruvate, which is 3 carbon, now it is broken down into two carbon. Those are the carbons. So from 3 carbon, they split, or 1 carbon breaks away, and they end up having only 2 carbon. Acetyl-CoA coenzyme. CoA stands for coenzyme. Simple. It will assist, or it will help pyruvate or acetyl-CoA in order to enter into the Kreb cycle. Don't forget, guys, all the reactions, all metabolic reactions in the body done with the help of enzymes. So acetyl-CoA assists the entry of the Kreb cycle. So before Kreb cycle to start, pyruvate must be converted into acetyl-CoA. As a result, here 1 carbon is broken. 1 carbon is broken. Another carbon is broken. Of course, in the presence of oxygen, carbon reacts with oxygen, simple to produce carbon dioxide. Simple to produce carbon dioxide. Two molecules of carbon dioxide. 1 carbon from this pyruvate. Don't forget we had 2 pyruvates, and another carbon from second pyruvate. Carbon, in the presence of oxygen, carbon will be oxidized. Simple, they will form 2 molecules of carbon dioxide. This is the beginning of, this is simple, the beginning of Kreb cycle. Still, we didn't start the Kreb cycle. This is the preparation of the Kreb cycle, where pyruvate broken down into acetyl-CoA and releases 2 carbon dioxide plus 2 NADH.
In the process of pyruvate oxidation, electrons are transferred to NAD, producing NADH, and a carbon is lost, forming carbon dioxide. The next stage is the citric acid cycle, also called the Krebs cycle. Here, acetyl-CoA will bind with a starting compound called oxaloacetate, and through a series of enzymatic redox reactions, all carbons, hydrogens, and oxygens in pyruvate ultimately end up as carbon dioxide and water. The pathway is called a cycle because oxaloacetate is the starting and ending compound of the pathway. For every glucose that enters glycolysis, the cycle completes twice, once for each molecule of pyruvate that entered the mitochondria. During pyruvate oxidation and the citric acid cycle, a net of 8 NADH, 2 FADH2, 2 ATP, and 6 CO2 are produced for each glucose molecule. In order to
So simple. Now we had 2 pyruvate, or we had 2 acetyl-CoA. We'll enter the Krebs cycle. Just to remind you, the word cycle series of events that repeat itself. As you can see, a starting material becomes the end material, or a start point becomes the end point. But of course, it is a cycle. It will continue wherever there is acetyl-CoA. Wherever there is an acetyl-CoA. Simple. Acetyl-CoA, we said it is 2 carbon. 2 carbon will join with this 4 carbon in front of you, as you can see. 2 plus 4, simple, it will form 6 carbon molecule. Simple, it will form 6 carbon molecule. Now this 6 carbon will lose as one of the carbon in the form of carbon dioxide plus NADH, forming 5 carbon. 5 carbon again will lose as one of its carbon in the form of carbon dioxide, forming 4 carbons. 4 carbons, of course, some changes its structure by the help of enzyme. Some enzymatic reaction will take place. We call isotopes. Those 2 are isotopes, different formula, different molecular formula, but different structures, releasing ATP and FADH2. Then back the cycle will start again and again. Because we had 2 pyruvates, so the cycle will, for each glucose, the cycle will turns, or the cycle will carry twice. Once for each pyruvate, or once for every acetyl-CoA.
Understand how the majority of the energy is produced by aerobic respiration, we need to follow the NADH and FADH2 molecules to the next stage.
Okay, so let's go back to the simple. Let's go back to our figure here. We enter the Kreb cycle. Each acetyl-CoA enters the Kreb cycle. Always keep in your mind we had 2 acetyl-CoA. Always keep in your mind we had 2 pyruvate, that means 2 acetyl-CoA.
Preparatory reaction prior to the Krebs cycle. The 3-carbon pyruvate first reacts with coenzyme A, CoA, to form a 2-carbon intermediate called acetyl-CoA and carbon dioxide, CO2. NAD+ is converted to NADH. Carbon dioxide is given off by the cell. Since there are 2 molecules of pyruvate for every preparatory reaction, it results in the production of 2 carbon dioxide molecules and 2 NADH molecules. Acetyl-CoA then moves to the mitochondrial matrix.
So they call it preparatory reaction, that means preparation of Kreb cycles. Before Kreb cycle, we said pyruvate must be converted into acetyl-CoA. Before Kreb cycle, coenzyme A forming 2 carbon intermediate called acetyl-CoA, and of course each pyruvate will releases carbon dioxide. So we had 2 pyruvates, so of course we will have 2 molecules of carbon dioxide. Beginning of the Kreb cycle, still Kreb cycle, we didn't enter the Kreb cycle. Those are preparatory reaction. Preparation of Kreb cycle. 2 carbon dioxide, 2 NADH molecules are produced. Of course, 2 acetyl-CoA moves into the matrix. Steps of Kreb cycle. Let's see how the cycle, or what are the steps, or what are the products in each
matrix. Steps of the Krebs cycle. The Krebs cycle begins with the 2-carbon acetyl part of acetyl-CoA combining with a 4-carbon molecule to form a 6-carbon molecule known as citric acid. The coenzyme A is released back into the mitochondrial matrix. Citric acid is then broken down to create 5-carbon molecules. During this process, a carbon dioxide molecule is released and a NAD+ molecule is converted into a NADH molecule. The 5-carbon molecule goes through a set of reactions. Another carbon dioxide molecule is released, and a molecule of NAD+ is converted into a NADH molecule. The process continues as a FAD molecule is converted into a FAD2 molecule and an ADP molecule is converted into an ATP molecule. FAD is another electron carrier, similar to NAD+ and NADP+. The final set of reactions occurs producing a 4-carbon molecule and converting a NAD+ molecule into a NADH molecule. The 4-carbon molecule combines with acetyl-CoA, and citric acid is generated. The cycle starts again.
So simple. For each acetyl-CoA or for each pyruvate, pay attention. Each pyruvate, by the way, before enters the Krebs cycle, will produce 1 carbon dioxide. Each one during the Krebs cycle producing the other 2 carbon dioxide. That means each pyruvate will release 3 carbon dioxide. So total, because we had 2 pyruvates, so total will be simple, 6 carbon dioxide per glucose. Simple. For each glucose, it will release 6 carbon dioxide. 2 carbon dioxide molecules from preparatory phase, which is the beginning or before the Krebs cycle, and 4 carbon dioxide molecules during Krebs cycle. So you will end up of having a total of 6 carbon dioxide molecules. Okay, let's go and see what about NADH and ATP molecules are produced per Krebs cycle. Remember, we saw that preparatory phase will release us 2 NADH molecule, 1 per each pyruvate. So during preparatory phase, each pyruvate, while it is converting into the acetyl-CoA, it will release NADH. So total you will have 2 NADH molecules in preparatory phase. Now, what about during the Krebs cycle? As you can see here, 1 NADH molecule, this is per pyruvate, of course. This is the second NADH molecules. And also third. So the total per pyruvate you will have 3 molecules of NADH. 3 molecules of NADH. So total per glucose you will end up of having 6 molecules of NADH. I will summarize this later for you. So during the Krebs cycle, 3 molecules and 1 molecule preparatory phase. So each pyruvate will release 4 molecules of NADHs. Here you have a new molecule. We have new molecule. It says FADH. FADH gives you FADH2 molecule, and here it says FADH is another electron carrier, same as NAD and NADP. That means those molecules, they will go to the electron transport chain, which will be our last stage or last phase of respiration, of aerobic respiration, in order to be converted into ATP molecules. So 3 NADH molecule during Krebs cycle, or total 6 NADH molecule during Krebs cycle, 1 FADH2 per pyruvate, or total will be 2 FADH2 per glucose, or total will be 2 FADH2 for whole cycle. Here, step by step, what happens in each phase. This is the summary of preparatory phase. Simple here, as you can see in front of you, pyruvate, in order to be acetyl-CoA, here we call it preparatory phase. This is preparatory phase. Okay, preparatory phase is before Krebs cycle. Before Krebs cycle. So as you can see, pyruvate converted into acetyl-CoA by releasing NADH and carbon dioxide. This is per pyruvate. So total times 2, so you will have 2 NADH and 2 carbon dioxide. So what happens in Krebs cycle? So what happens during the Krebs cycle? This is what is known as the Kreb cycle. Of course, we are in matrix of mitochondria. Krebs cycle, matrix of mitochondria. So each acetyl-CoA here enters the Krebs cycle. We are interested in number of NADH and FADH2 molecules. So here, this is your first NADH, second NADH, and third NADH molecules. This is per, we are taking per acetyl-CoA. So 3 NADH molecules. Let's count the carbon dioxide. We have 1 carbon dioxide. We have one CO2 over here. Okay, so one carbon dioxide over here. And let's check the FADH2. One FADH2 per cycle. This is per cycle. So 2 carbon dioxide, 3 NADH molecule, and 1 FADH molecule. And of course, as you can see here, they just simple generate or just simple they release 1 ATP molecule. They just release or produce 1 ATP molecule. As I said, we are interested in number of the molecules in each phase, or number of, or the simple reactant and product of each phase. Phase 1 glycolysis, phase 2 Krebs cycle or aerobic respiration. We are not interested as the name of the molecules or what do they form. Simple, just what do they produce at the end of each phase or at the end of the each cycle.
2 molecules of pyruvate are formed during glycolysis. Therefore, it results in 2 turns of the Krebs cycle for each glucose molecule. The net yield from the Krebs cycle is 6 carbon dioxide molecules, 2 ATP, 8 NADH and 2 FAD2. 10 NADH, 8 from the Krebs cycle and 2 from glycolysis, and 2 FAD2 move on to play a significant role in the next stage of aerobic respiration, the electron transport chain. Aerobic respiration, Krebs cycle glycolysis has a
So simple.
Practice.
Simple,
what are the
guys.
products of the Krebs?
Let's go and see the Krebs cycle, or what are the products of Krebs cycle. So of course we will start with 2 acetyl-CoA, right? This is the reactant of Krebs cycle, 2 acetyl-CoA. Simple. In order to, or during the Krebs cycle, it will generate or it will produce 4 carbon dioxide plus 3 NADH plus make it 6 because we are started with 2. So 6 NADH, 2 FADH, plus 2 ATP molecules. This is only, or this is simple in Kreb. This is simple in Kreb cycle. This will take place only in the Kreb cycle. So if you go and check, or if you go and count preparatory phase and Kreb cycle, preparatory phase plus, you will add all together, preparatory phase plus Kreb cycle. Here we can just write, we start with 2 pyruvates, simple. It will just release 6 CO2, 6 carbon dioxide. We are doing, guys, the total preparatory phase and Krebs cycle both together. So 2 pyruvate will give me 6 carbon dioxide plus 8 NADH. Why 8? Because I had 2 in preparatory phase and 6 in Krebs cycle. So the total will be 8. Total will be 8 plus 2 FADH2 plus 2 FADH2 or FADH plus 2 ATP molecules. So this is the total of preparatory phase and Krebs cycle. Yeah, questions? Guys, you are responsible just to know the number of molecules in each phase. Simple example, you will have a multiple choice question. What are the total number of NADH are produced in Krebs cycle? Krebs cycle, here it is. 6 NADH in Krebs cycle. Preparatory phase and Krebs cycle, so the total will be 8 NADH. 8 NADH. One important thing also, FADH2 or FADH equals 2 ATP molecule. Remember I gave you last time NADH is equal to 3 ATP molecule. So now each FADH is equal to 2 ATP molecules and NADH equals to 3 ATP molecule. So by tomorrow or by Wednesday or the last phase, we are going to see what happens to these NADH and FADH2. They have to be converted into ATP. They have to be converted all into ATP. Okay, so this is what you are responsible for. Glycolysis, what do they start? What do they produce? Krebs cycle, what are the starting materials? Simple, the Krebs cycle starts with 2 pyruvate. 2 pyruvate gives you 6 carbon dioxide, 8 NADH, 2 FADH2 and 2 ATP molecules. So here, if you want, guys, you can simply add, just go back and add glycolysis. Glycolysis, you start with glucose, gave us 2 pyruvates, plus 2 ATP plus 2 NADH. So at the end of the lesson or at the end of the aerobic respiration, we are going to add all the products. So here pyruvate, we are interested in ATP and NADH. So 2 ATP, 2 NADH plus 6, so total, plus 8. Total till now we have 10 NADH. 2 plus 2, we had 4 ATP and 2 FADH2. This is the total till Krebs cycle. So keep this, I will give you a summary when we... Yep, let's start. Just to remind you, electron transport is the last stage of aerobic respiration. It takes place inner membrane of mitochondria. ATC takes place inner membrane of mitochondria. Simple in the matrix. Krebs cycle takes place in the matrix. ATC, electron transport chain, will take place in cristea, or inside the membrane, or inside inner membrane of mitochondria, which is known as cristea. Okay, so here this is the inner folded membrane of mitochondria, which is known as cristea, where electron transport chain, or the last stage of aerobic respiration, will take place.
When glucose is oxidized during glycolysis and the Krebs cycle, the coenzymes NAD+ and FAD are reduced to NADH plus H+ and FADH2. In the mitochondria, the electrons from NADH plus H+ are transferred to the electron carrier proteins, and the protons are transferred across the membrane. As the electrons move from cytochrome to cytochrome down the electron transport chain, more protons are carried across the membrane. Cytochrome C transfers electrons to the cytochrome C oxidase complex. Protons are also transferred to the outside of the membrane by the cytochrome C oxidase complex. The cytochrome oxidase complex then transfers electrons from cytochrome C to oxygen, the terminal electron acceptor, and water is formed as the product. The transfer of protons generates a proton motive force across the membrane of the mitochondrion. Since membranes are impermeable to ions, the protons that re-enter the matrix pass through special proton channel proteins called ATP synthase. The energy derived from the movement of these protons is used to synthesize ATP from ADP and phosphate. Formation of ATP by this mechanism is referred to as oxidative phosphorylation.
So, as we saw in the video, here when the NADH and the FADH2 are releasing their hydrogen into the outer space of matrix, so here it will be concentration gradient, or it will be high concentration of hydrogen. As you know, hydrogen will diffuse simple from high concentration to low concentration through the ATP synthase channel. From its name, ATP synthase channel, main purpose or main function of this channel is to produce ATP synthase channel. So the main purpose, main function of this channel is just to add phosphate to ADP, or just to phosphorylate adenosine diphosphate into adenosine triphosphate. So the aim of NADH and FADH2 is just to produce, or just to release protons, which are H+, into outer space of the matrix. Then those H+ will diffuse into the matrix. By this, they are going to phosphorylate, or they are going to add phosphate to ATP, forming adenosine triphosphate. So to understand the whole process, first let's make a summary. Glycolysis. Simple glycolysis produces 2 ATP plus 2 NADH, 2 ATP plus 2 NADH. Kreb cycle produces 8 NADH, 2 FADH2, 2 ATP plus 6 CO2, 6 carbon dioxide. We are just summarizing the stage, each stage of respiration and what do they produce. ETC is simple, it will produce 32 ATP plus 6 H2O. Now, some of you will ask why 32 or how 82? So total per glucose, total ATP per glucose equals 36 ATP molecules. That means each glucose will yield 36 ATP molecules. Here, how 32? Simple. 8, 2 plus 8, 10 NADH, and you know that each NADH equals to 3 ATP, so total will be 30. We have FADH2 will release us 2 ATP, so total is 32 ATP simple from ETC. And we had 2 ATP from glycolysis and the other 2 ATP from Krebs cycle. So 32 plus 4 simple, the total will make 36 ATP per glucose. So pay attention for glycolysis, Krebs cycle, ETC. Each stage has different number of ATP molecules. Each stage has different number of ATP molecules. ETC, simple, 10 NADH equals to 30. Why? Because each NADH is almost equivalent to 3 ATP and FADH is equal to ATP. So you will have 32 from ETC. 32 ATP simple from ETC. Yes. If you write the equation of cellular respiration, we said glucose plus 6 carbon, 6 oxygen it gives 6 carbon dioxide plus 6 water plus 36 ATP molecule. And here what we got from each stage. We start with glucose, oxygen found in the mitochondria matrix. It yields to 6 carbon dioxide, produces 6 carbon dioxide over here, 6 carbon dioxide as a product, 6 water here, so I have 6 water, 6 carbon dioxide, and the total, if you add all the ATPs that are produced from each stage, you will end up of having 36 ATP molecule per glucose. This is one glucose will yield to 36 ATP. So this is the summary. This is simple, the summary of number of ATPs per each stage of aerobic respiration. Per each stage of aerobic respiration.
Respiration Electron transport is the final step in the breakdown of glucose. It also is the point at which most of the ATP is produced. High-energy electrons and hydrogen ions from NADH and Fed2 produced in the Krebs cycle are used to convert ADP to ATP. Electron transport and chemosmosis in cellular respiration are similar to these.
So here, of course, here they told you, or they show us the steps. Simple, ETC will start with NADH and FADH2. They will start with NADH and FADH2. Of course, we have 10 NADH and 2 FADH molecules. Simple, they will donate the hydrogen, or they will donate the protons, and they will be converted into NAD and FAD. It can be reduced again, so oxidation reduction takes place. High concentration or H+ ions are pumped into membrane space, creates increasing concentration of H+ ions in the outer membrane. So as a result, high concentration of hydrogen ions, simple, will diffuse down concentration gradient. That means it goes from high concentration to low concentration, of course, through ATP synthase channel. So those hydrogen pluses or H pluses, they will diffuse into the matrix through ATP synthase channel. From its name, ATP synthase is responsible for synthesis of ATP. As H pluses moves through the ATP synthase, ADP is converted to ATP in the mitochondrial matrix. Oxygen is the final stage of the acceptor. Of course, it will bind with hydrogen, simple, to form water molecules. Simple to form water molecules. Of course, as a number, it will be 6 oxygen, 6 water molecules from aerobic respiration. Overall, as ETC, here ETC produces 32 ATP, and 2 ATP from glycolysis makes 34. 2 ATP from Krebs cycle, simple, the total will be simple. 1 molecule of glucose yields 36 under ideal condition, that means simple during aerobic respiration. Simple during aerobic or ideal condition when oxygen is present.